So let's do a quick recap of what we did uh, last time. Okay, so we talked about innate immunity, which refers to immunity that a person is born with. These are basically hardwired into the genome. And as soon as the baby is born, they would have the innate immunity. So the best kind of defense is to keep things out. So we have a physical barrier, right? Such as skin, mucus, membrane, and a normal flora, right? So these help keep things out of the body. We also have chemical barriers, which include things like low pH, lysozyme, right? These are antibacterial substances that could be found in tears, saliva, and sweat, and it helps cleanse exposed surfaces, right? But when they do make it into the body, then we have inflammation as a way to kind of recruit more white cells and plasma protein to help clear the infection and help initiate the healing process. Okay, so in inflammation, we talk about mast cells, right? We talk about histamine, which will cause the blood vessels to become bigger. It will increase the permeability of the blood vessels, right? And we talk about the characteristics, the hallmarks of inflammation, which include redness, warmth, pain, as well as swelling. As the inflammation is happening, there are also cells like phagocytes, right, which will help eat up the invading pathogens. If the pathogen express PAMS, PAMPS, it will be recognized by the phagocytes and they will be eaten up. Okay. And also, there are these NK cells, natural killer cells. Right? So the NK cells, they are going to attack virus-infected cells. And also, with some limited capability, they are able to clear tumor cells as well. Okay, so that's sort of where we left off. Okay, maybe just a few things here to write down. They recognize PAMs, right? The pathogen associated molecular patterns, and they're gonna go eat them up. They can also secrete colony stimulating factors, right, to stimulate white cell production in the bone marrow. So you can call for backup, right? You can get more white cells coming out of the bone marrow. And again, NK cells, they kill virus infected cells. Okay, and tumor cells. Okay, so that's everything that is happening with the innate immunity so far. All right. So the next strategy that is being used by innate immunity to help fight off infection and pathogens are these things called complement proteins. Okay, complement proteins. They are plasma protein that are made primarily in the liver. And when you are not fighting a pathogen, these proteins are not turned on. Okay? They will circulate in the blood, right? But they are in an inactive state. When you are fighting a pathogen, then you have to turn them on one by one. Right? So it's a series of chemical reactions, and there are different pathways to turn them on. Like obviously you don't have to memorize these things, but you can see one thing leads to the activation of next. Right? So these blue ones get activated, and then it's gonna activate the C3, which then go activate like C3A, C5A, so on and so forth. It's like a domino effect, right? Remember those coagulation pathway that we talked about, the prothrombin and the fibrinogen. It's kind of like that, right? Where the activation of one thing, it's going to lead to the activation of the next. Okay, so through a series of chemical reactions, multiple complement proteins will be activated. Now, depending on which complement proteins are activated, the actions are going to be different, the outcome are going to be different. But generally speaking, they will help synergize another non-specific defense. To synergize is basically to enhance, right? To make it better. So I'm going to give you 3 examples of how complement protein works. First example, it's going to help recruit more white blood cell. Okay? Recruit more white blood cell. Now, isn't that the function of inflammation, the purpose of inflammation, right? To help bring more white cells, right? And that is also the function of the colony stimulating factor, right? Okay? Think about it. The colony stimulating factor from the white cells, it's going to go to the bone marrow. Okay? So the bone marrow will make more white cells. And suppose I'm a white cells that is freshly made. I came out of the bone marrow. How do I know where to go? The body is so big. How do I know where the infection is, right? So it turns out the complement proteins, they will leave a chemical trail for the white cells to follow. Okay? It's kind of like, the Hansel and Gretel story, right? Where they leave some breadcrumbs so that they could follow the trail, right? And these chemical trails will attract the white cells, the newly made white cells or white cells that are already in the body. They will attract them to the site of infection, and therefore able to increase the recruitment. That is one function of the complement protein. Now, the second function is to help tag the pathogen. Okay, so look at this white cell. This white cell came out from the blood vessel. Okay, it followed the chemical trail. It went to the site of infection. It squeezes itself out of the blood vessel. But how does it know which one is the target? Right? Sure, they can check the PAMs, okay, but it's hard to identify it. So that's why the second function of complement protein is that it will coat the target cell. Right? By coating it, it makes it easier for phagocytes as well as antibodies to identify them. It's like this game, right? You might have played this before, or like your kids, right? It's called Finding Waldo, right? Like you have to find this guy in the picture, right? So all these people represent cells that are encountered by like a white cell, and the white cell is trying to find like where the Waldo is. It's trying to find where the target cell is. Like which one am I supposed to eat, basically, right? So like you're gonna find it, but it's gonna take some time, right? Okay. So what the complement protein does is the complement protein is basically going to put a big red circle around the target. And so if you see a picture like this, immediately you're like, oh, there's the thing I'm supposed to attack, right? There's the thing that I'm supposed to eat up, right? So essentially the complement protein put a big sign on the pathogen and say, please eat. So highlights the target, makes it easier to identify. Okay. So creating a chemical trail for the white cells to follow, tagging the target so that it is easier to identify. And the last function of the complement protein is that they are going to form these things called the membrane attack complexes. Right? Essentially, they assemble into this structure, which will punch holes into the bacterial cell wall. Right? And when you have the bacterial cell wall punctured, basically the bacteria will leak out all of its content and it dies. Okay. So there are many different functions of complement proteins. I only gave you three, as examples. Right? The first two, the chemical trail and the tagging, those are kind of passive ability, right? Whereas the third one, it's more like active attacking of the pathogen. Okay. So let's take a look at these poll questions. Okay. This is a misconception for a lot of people. But PAMS, right? PAMS, if you forgot, it stands for pathogen associated molecular pattern. Okay. So it's going to be on the pathogen. Right? It's not on the phagocyte. The phagocytes, which are like the neutrophil, right, macrophage, these are the cells that would recognize the PAMs, and then they would attack the pathogen. Right? These are the PAMs are basically like dangerous signals on the pathogens. And when the white cell sees these dangerous molecules, they know to attack it. They know to eat it up. Okay, so PAMs are on the pathogens. Okay, they are markers for white cells to identify and to destroy. Let's try another one. Okay, so as I was saying earlier, the NK cells right here, the NK cells are the ones that are going to be killing virus infected cells as well as tumor cells. So if a person is lacking NK cells, they will have a higher than normal chance of developing malignant tumors, right? Okay.
Can you explain the other cells too, please?
Can I explain what, sorry?
The function of other cells a little bit.
Yeah, so like some of it, like for example, B cells, we're gonna learn in a lot more detail today, but previously we wrote down, I think it was here, was it this one? Let's see if it's this one. Oh, even earlier, I think. Previously, we wrote down that the B cells, right? B cells, they make antibodies, right? Um, and as for the other ones, we haven't learned this yet, so I'm not sure why people choose that one. Macrophage, so make antibody. Macrophage, they perform phagocytosis. Okay. Anybody remember basophil? What does basophil do? What does it trigger? You gotta take a look at your notes, right? That table that we did. Inflammation. Okay. So basophil and mast cells, they both play a role in inflammation. The difference is mast cells, they're more for like localized type of inflammation, right? Okay, so there you go.
Thank you.
Yeah, it's probably a good idea for you, like all of you, to write down all the cells that we've learned and you know, classify them as well as write down their function. I have them on a sheet. Um, it's going to be quite useful. All right. Now, next question.
What do you mean by the membrane attack?
Membrane attack complex. So that's one thing. That's one thing. Like it's the three word is one thing. It's not just membrane attack, but it's the membrane attack complex. So what form these? All right. Thank you for choosing the answers. Let's go back here. Look. The complement protein, right? They form the membrane attack complexes, right? These are things that will punch holes into the bacteria, causing them to leak all over the place and die. So membrane attack complexes are formed by complement proteins. All right. So, like I said, like I understand there are a lot of things going on in the lecture, but you know, if you could organize the information in the study guide, it would be useful for you, right? So like there is a table, right? The barriers to entry, like write down what the purpose is. Purpose is to keep things out, and then description. We have the physical and chemical barrier. Okay, so you should go through these things, right? So it's gonna be all organized in one place. Okay, I think there is another maybe not here, but like I remember there's another maybe in another study guide there are cells on one side and then there are functions on the other, and you can match them up, right? So please do that as well. Okay, all right, back to the PowerPoints. Now the fifth mechanisms for innate immunity is fever, right? If everything else is not quite, working, you're not really able to clear the infection fast enough, then your body's gonna turn up the temperature, right? And that's basically what fever is. Normal body temperature is about 37 degrees Celsius or 98.6 degree Fahrenheit. And there is a part of the brain which controls body temperature. It is called hypothalamus. Okay, and the hypothalamus will release a group of chemicals called prostaglandin that will cause the temperature to go up, basically. Okay, so the hypothalamus is right here. Okay, it's right here. We will learn about the hypothalamus in lecture 6, okay, when we do the brain parts. But what does the fever do? The fever is going to help enhance the immune defense by increasing enzymatic activity. Enzymes are proteins that are carrying out chemical reactions, right? And a lot of what's going on during an immune response are basically chemical reactions. Think of the complement proteins, where one thing leads to the activation of the next. Those are essentially chemical reactions. And by turning up the temperature, you can increase the reaction speed. Therefore, you can activate the complement proteins faster. Okay, so e.g., activate complement protein faster. Speeding up metabolism. Right? Metabolism in general are reactions that are going on in a cell. So if there is any kind of wound healing, right, that higher temperature might be able to do it for you. Increase the rate of tissue repairing and phagocytic activity. It's like pressing the fast forward button, right? When you watch a movie. Everything just goes a little bit faster, with a fever. And also the higher temperature can also reduce pathogen activity.
I have a quick question.
Yeah.
So when you get like too high of a fever, it can cause like seizures and a bunch of other bad stuff. So how does your body like know how to kind of regulate the fever?
Yeah, it doesn't. That's the problem, right? So like a little bit of fever is okay, but sometimes it gets out of control. The fever is not able to get rid of the pathogens along with all the other innate immunity. And your body kind of go into this overdrive, and it caused the temperature to go beyond like well into like the 40s, then it becomes very dangerous, especially if it's for kids, right? So in those cases, that's why we need to bring the temperature back down, right? But generally speaking, like if you are, running at a low fever, like a 38 or 39, some people say it's okay, just, let the body does its thing, because it's part of the healing process. But anything above 40 is, probably should, take the drugs and, get medical help. All right. So that is that. Are we okay? Any questions? I feel like, I I don't know, I can feel your frustrations, right? It seems like the first quiz was not as I thought it went pretty well. Okay, looking at the marks, like nobody failed. That is a very good thing, good sign. and the classification is good. But if you felt like it was harder than expected, I don't know. Give me some feedback. Send me an email. Okay, let me know why it was difficult, and if you have suggestions, let me know. I'll take it into consideration.
Professor, maybe you can just give us some application example together with the topic so we have an idea about it.
Yeah, it's kind of my fault, sort of, because I totally forgot to take up these questions with you. I was supposed to, but then I forgot. So maybe that was part of the problem.
No, I
Uh,
even read those questions. I searched them on the Google. I did went through some of them as well
yeah.
for both of these questions. I went through all these lines. Like I was able to get the material from the internet for
Mhm.
ECE,
Yeah.
but it's much better when you explain. It's much clearer.
Right. So I try to remember them, right? So like there was a question about congestive heart failure, right? Do you guys remember that on the test? Some of you
Yeah.
probably had that, right? And then I forgot to went through the application questions here. But I'll try to do that before the next quiz, next test. And more importantly, you have to do them as well, right? Like you gotta. Some people look at the application question and then they see that there's no answer in the study guide and they're like, oh, it's okay. I'll just like maybe not do them. But these are the questions that will show up, right, on the multiple choice and whatnot. Okay.
Professor, I did that, but the thing is, it's difficult to manage. Like in the exam, it was saying if the left side stops. So I was thinking first it may be causing dehydration because blood is not moving, and then I thought no, because the blood is not coming from the back, then it could be like
Yeah.
in the lungs it is more blood.
So maybe I
So,
explain this one right now, okay? Just
yeah.
since we're here anyway, okay? So again, there was a question about congestive heart failure on the quiz, and you don't really have to know what it is. That's the whole point, right? Like it's application. You're supposed to extract the information on the spot, right? So the question tells you it happens when the ventricles fail to empty its content. So let's say we have the right atrium, right ventricle, left atrium, left ventricle, right? So let's say if it's happening on the right side. So if the right side is not emptying, it's not emptying, right? That means the right atrium is also not going to be able to empty. Think of like a traffic jam. If there's a car accident somewhere, then everything before that is going to be blocked, right? The traffic is going to slow down. So if the congestive heart failure happens at the right ventricle, the blood is not emptying, then the blood is also not emptying from the right ventricle. And then you have to think about where does the blood come from for the right atrium. What vessel brings the blood to the right atrium? Well, it is exactly right, the vena cava, right? So the blood is going to be also pooling in the vena cava, right? And as a result of that, you are not draining, you're not returning all the blood from the body, right? And so you are likely to have blood pooling in the body. And because of gravity and all that, the blood is going to be pooling in the legs. And we know if the blood is pooling in the legs, that increases the blood pressure and it's going to be pushing the blood out with greater forces. And so you will have swelling in lower limbs. Okay. Whereas if it happens on the other side, then this is from lungs, right? From lungs. So instead of having swelling in the lower limbs, if the congestive heart failure happens on the left side, then you will have, edema. You will have swelling in the lungs. You will have pooling of fluids in the lungs. Okay, so that is the answer.
Sir,
Mm-hmm.
what does that R mean? That was a question on the.
Yeah, so this here is ventricular contraction, right? Let me show you the slide. I think we wrote it down one time during the test. No, during the lecture. let me just show you quickly. Right, so there is a slide here, right here, right? So look, this little peak, right? I remember in the lecture I wrote, this is atrial contraction, right? And then I say there was a little lag time between the signal getting from the AV node to the Purkinje fiber, and then this is ventricular contraction. All right. So I mean, I'm not trying to defend myself, but still, I want to tell you that most of the questions, the answers are somewhere in the PowerPoint slides. Okay. And that's why I encourage you, right, to, like, you know, really spend time going through the study guides as well as the slides to familiarize yourself with it. You might not even remember all of the details, but at least you will know where to look, right, when you see a picture of like, for example, like if you see the ECG graph on the test, I think there was a question like that, then you can find this slide, or you're like, oh, I remember seeing it. Okay, and then you just have a quick reference, right? I don't know. It's
Professor, I have a question.
yeah.
You say the next bulb is when it is resetting the heart is resetting.
At the very end of the ECG,
Very
yes.
end. Yeah.
Yeah.
So what is in between, like the big peak and a small bulb, like?
Yeah, like I closed it already, but you're talking about like this part here?
No, the next one after it.
Oh, the one that goes like this at the end? This part?
Yeah.
Yeah, that's just like resetting. The whole thing is resetting. I mean, it's more complicated than that, but
Oh.
you know, to keep it simple, it's just, you know, resetting, waiting for it to.
If we remove the AV node from the heart.
That's right. So if you, there was also a question where asking you what happens if you remove the AV node, but we do not disrupt the signal, right?
Yes.
It's still able to go to Purkinje. So without the AV node, then there will be no pause. Then the whole heart would, all four chambers would just contract at the same time.
Will it
Right.
not be not contracting, because AV node is basically passing the signal from?
Yeah, but then the question says without disrupting the signal transmission, right? That's the key part, right? You're right. If you just remove the AV node, the signal would not be able to get on, right? But the question,
Yeah.
I believe, also says without disrupting the
Yes.
transmission. Yeah. So sometimes they're a little bit tricky like that. Anyways, don't feel discouraged, guys. I honestly think like the results are not too bad. Like I would be alarmed if a lot of you like fail, but you did not. But hopefully after this quiz, you will see what it's like. I mean, it's pretty similar to the first semester, right? What to expect. It's just more content, I suppose. But yeah. Anyway, let me know if I can help in any other way. We should move on. So the next thing we're gonna do is adaptive immunity. Okay, adaptive immunity. Remember, you were born with innate immunity and you are equipped with adaptive immunity. But adaptive immunity requires exposure, right? You are not going to be able to turn it on until you encounter something foreign. That's why it's not the same throughout your lifetime. It's able to evolve, right? And it's something that you have to acquire. The whole point of adaptive immunity, as you will see, is to make antibodies. That's what this thing is. This Y-shaped things, these are antibodies. The antibodies are the ultimate weapon of the body when it comes to fighting pathogens. If the antibodies don't work, then there's nothing else your body can do. You have to, like, you know, rely on medication to help, you know, get rid of the pathogens and whatnot. Okay, so the whole point of adaptive immunity is to make antibodies. So if innate immune mechanisms do not quickly eliminate an invading pathogen, then you have an established infection. And which is why you need to create the adaptive immunity. Okay. So if you compare the innate versus adaptive, the innate is more general strategies. It's not designed to fight a specific thing. Okay, so they're good for like weak everyday encounter pathogens. Like if you eat your sandwich without washing your hands, you are consuming some pathogens along the way. Those are okay. Like your innate immunity is going to be able to take care of it. All right. However, if you consume some E. coli or something, or salmonella, right? Then the innate immunity might not be equipped to deal with those more vicious infection, and that's when you have to bring out the adaptive immunity to create a tailored solution for whatever is infecting you. Okay, so innate immunity is non-specific; it's broad spectrum. Whereas adaptive immunity is tailored, is very specific to the invading pathogen. Okay. The characteristics of adaptive immunity lies in the ability to distinguish self versus non-self. They are able to identify from a bunch of cells, like which one stands out, which one does not belong. That's going to be the one that needs to be attacked. Okay. Sure, the macrophage can do that by recognizing PAMs, but there are some pathogens that are good at hiding those things. And so you need adaptive immunity to kind of, identify them and create some kind of immune memory so that in the future, when the same thing comes attack you, it's all right. You remember attacking it previously. You have knowledge of that thing, and so you're able to create a response that's much faster and much stronger. Okay, future encounter of the same pathogen will always be stronger and faster. Right? That's why sometimes we need to have a booster when we get vaccinated. Right? Yeah, it basically prime your immune system for reoccurring infections. The adaptive immunity is also high in specificity and at the same time very diverse. So the best way to visualize this is to think of a set of screwdrivers. If you have this set of screwdrivers, each screwdriver is very specific in terms of the kind of screws that you can turn, right? Like if you have the I don't know what they're called, but like if you have the one that is a cross, then it's good for tightening the cross ones. Okay, if you have the one that is like a hexagon or whatever, right, the hex key, then you can tighten those kind of screws. Okay, each one is very specific. But if you like I cannot use the one that is a cross to tighten something that looks like that, right? Okay, so each one is very specific. But if you have the whole set, then you can essentially tighten any screws. Yeah, it's very diverse because you have the whole set. So when it comes to adaptive immunity, each antibody that you produce is generally good for one type of pathogen. The anti-chickenpox antibodies that you make are only good for chickenpox. However, when you consider the entire adaptive immunity, you have the potential to make antibodies for any kind of pathogens, not just chickenpox. You can make it for hepatitis. You can make it for, E. coli. Okay, so there is the potential to make billions, to recognize billions of different antigens when you consider adaptive immunity as a whole. Okay. Now, some of you might be a little bit confused because sometimes I say pathogens, sometimes I say antigens. Like, what is the difference, right? Remember, the antigens are things that are on the surface of a cell, right? So a pathogen is going to have different antigens on it, and these are going to be foreign to the body, and that's what we are making antibodies to recognize. Okay, so as I told you earlier, central to adaptive immunity is the ability to create antibodies. That's the whole point. The whole point is to make the antibodies to attack the pathogens. Okay, just like the screwdrivers, the handle of all the screwdrivers are pretty much the same, right? What makes one differ from the other? It's the tip, right? The tip is going to dictate what kind of screws you can screw. And here, in the case of the antibody, it's the same thing. First of all, it's a Y-shaped protein, and then there is the handle, which are the same for all the antibodies. We call that the constant region. And then the arms of the Y, that is the variable region. Okay, so that's going to determine what kind of shape you can grab onto. We already learned about this a little bit, right? Like in lecture 2. If you make an anti-A antibody, they will recognize the A protein. Right? So in this case, if you take a closer look at the picture, this virus have like little hexagon on them, right? Little hexagon, or pentagon rather, sorry. So the antibody will recognize those particular shape. And this particular antibody on the left will not work for the red bacteria here, because this one doesn't have the hexagon. It has the little bumps. Okay, so every time you create a new set of antibodies, it will be specific for the kind of pathogens you are trying to fight off. Okay, and if you are able to identify like a unique shape on the virus, on the bacteria, and make an antibody for it, then that antibody is particularly good for fighting off that antigen, that pathogen. All right. Any questions before I tell you more about antibody? So sorry, go ahead, Mohammed.
Antigen is on the pathogen.
That's right. So antigen is molecules that are on a cell, okay? Not just the bacteria. It could be on our cell as well, right? Remember when we talk about like blood type, the A, B, O, those are antigens, okay? But you have to remember antigens are on all cells, and they don't always have to do with blood type, right? You can just have like some sugar molecule on a bacteria. If it's a sugar molecule that we don't have, then it's a foreign antigen. Okay, so remember, guys, antigens are not good or bad. They are just things that are on the cell. Okay, but when they are found on a pathogen like a bacteria, your body is able to distinguish whether it's foreign or not. That's what the self versus non-self is, right? It's like, oh, that bacteria has some funny molecules on it that I've never seen before. That's foreign. We should make an antibody to attack it. Okay. So how does it work? How does antibody work? And I'm going to go through like 3 ways to show you how antibody works. So first, antibodies, they react with viruses and toxins to neutralize them. Okay. So let's say there is I don't have a picture here, but like, you know, I told you previously, like if this is the virus and then this is your cell, C-E-L-L, the virus is going to bind to the cell, right? Okay. Oh, sorry, the picture is down here. I didn't even see it. Sorry. So here is like your cell, and then these are the viruses. Usually the virus will go into your cell and hijack the cell machinery, right, to make more viruses. But if we have antibodies around, the antibody will bind to the viruses and they cannot touch our cells. They cannot go into our cells. Therefore, you neutralize the virus. it's also possible to neutralize toxins. Okay? Toxins are poisons that are made by bacteria. Okay, these things you can cook your food 100%, right? Well done, cook it and eat it, and you can still get sick from it because the poisons are not going to be broken down by the heat. Just because you killed off the bacteria, the poison might still be around, right? So then if you have some toxins in the body, if you get bitten by like a poisonous snake, the antibodies will be able to bind to these toxins and again, neutralize them, preventing them from harming your cells. So that's the first function of antibodies, neutralizing toxins or viruses. Also, it's able to bind to the target and cause them to clump together. We learned this already in lecture 2, right? The agglutination, right? When you have mismatched blood type, the antibody and the cells clump together. This is the exact same thing. But instead of clumping the wrong blood cells together, these are clumping the pathogens together. Okay, so these like green little ovals here, the antibodies are binding to them and it makes them clump together and form these big chunks, and it makes it easier for the macrophage to eat them. Okay, so neutralize toxins, neutralize virus. They form these big chunks, big clumps, and make it easier for phagocytes to eat them. And also, it's able to work with complement. Okay? So complement is part of the innate immunity, right? Remember? Complement proteins, the one that leaves the chemical trail and form those like MACs, right? So it turns out the antibody, even though it's part of adaptive immunity, is able to work together with the innate immunity. It helps activate the complement and form these membrane attack complexes. And it also enhances phagocytosis. So it's not like innate and adaptive are like, independent departments, if you would. They actually work with each other. They have the same goal, to clear out the infection as quickly as possible. Okay. Now there's one more word that might make it even more confusing. Okay. So like this is the pathogen, okay? Pathogen. And on the pathogen, they have different shapes, right? They have different shapes. Okay. These are called antigen. So far, so good? Pathogen, antigen. But when you make the antibody is actually quite small. The antibody is quite small. It binds to not the whole antigen, but parts of the antigen. Okay? And that part of the antigen is what we call like an epitope. Okay? Epitope. It's a particular part of the antigen. So like this whole thing is the antigen, and then you would have like the pathogen, right? Like the bacteria. This is not to scale. And then you can make antibodies that recognizes different parts of the antigens. Some are stronger. You can see it binds to like two, like binds with both arms, and then some are only binding one arm. And so being able to identify which part of the antigen, which epitope is most immunogenic is going to help create a stronger immune response. Okay. So, let's help you understand this by, doing an exercise. I'm gonna describe an animal part, and then you're gonna tell me what animal I'm talking about, okay? You can unmute and tell me, or you can just think about it in your head, okay? So for example, if something has a long nose, right? What animal am I talking about? Hopefully you're thinking about elephants, right? Okay, next one. If something has a long neck, what am I thinking about?
Giraffe.
The giraffes.
Exactly, right? So what about I say something that has a tail? Then what am I talking about?
Dog.
Yeah, it could be a dog, right? Could
A
be
lion.
could be a lion, could be anything, right? Okay. So my point is this. If you are able to identify a unique defining feature of a pathogen, like the trunk of an elephant, like the neck of a giraffe, then you are going to be able to create an antibody that is more effective in identifying it. In other words, when we try to identify the targets with antibodies, we don't want to find the tails, so to speak, because every animal has a tail, right? It's not a uniquely defining feature. So when we try to make antibodies, we want to find something that's unique for the pathogen. Okay, the more unique it is, typically, the stronger the antibodies are going to be. So I don't want the tails of the pathogen because all pathogens probably has that tail and it's not going to give me a good immune response. Okay. So let's pause for a second. I have told you a lot of things, and I understand this can be overwhelming. So if you have the study guide, maybe we can go here and fill in this chart here, this table. This is a nice summary that compares innate and adaptive immunity. Okay. So remember, innate immunities are immunity that you are born with. Okay. And they work right away. So the response time for innate immunity is immediate. It works right away. You touch a dirty surface and then you eat chips with your hands. As soon as the pathogens goes into your mouth, the innate immunity will immediately help you clear those pathogens. But if you come down with something much more serious, like if you catch COVID or something and you were not vaccinated, then the adaptive immunity will take some time to make the antibody. So this is not immediate. Okay, it takes time to make antibodies. Right? What about specificity? The innate immunity, again, in case if you are confused, innate are the stuff that you're born with, like the intact skin, like the ability to create inflammation and fever, and the complement protein. These are non-specific protection. Like the skin is good for keeping most pathogen out. It's not creating a barrier that only works to keep like E. coli out or something, right? It's like a broad spectrum strategy. So this is non-specific. Whereas adaptive immunity is specific. Okay? Again, it is a tailored strategy for a specific pathogen. Okay, I keep on saying the antibodies you make for the green virus is only going to be good for the green virus. It's not going to work for the red bacteria, so to speak. Innate immunity, how antigens are detected, right? Recognition of PAMs. Right? That's the way that we talk about. If the pathogen has these, dangerous signals on them, the pathogen associated molecular patterns, then the innate immunity will recognize them. The white cells will identify them, attack them, eat them up, and hopefully all is good, right? As for adaptive immunity, it basically use something called gene shuffling. Okay. Long story short, when you shuffle your genes, because genes are DNA, right? DNA contains the instruction to make the proteins. When you shuffle the genes, the shape, the configuration of the protein that you make will vary, will change. So you have a set of genes that are dedicated for adaptive immunity. And when you shuffle them, there are literally billions of different combinations that you can make. Okay. Billions of combinations. So that's why you are potentially able to recognize any foreign antigen, right? And you are potentially able to make antibodies for all of them. The problem is, like, are you able to make them fast enough, right? Like, some diseases are very very virulent, right? Like it causes you to become very ill very quickly, and your body is not going to be able to create an antibody fast enough before the disease kills you, right? So it's not that you cannot make the antibody, but you cannot make it fast enough in some cases. And if something is very transmissible, right, like you spread it very quickly, that's also a problem. Like, if you get COVID and then before your body makes the antibody to make you feel better, the whole time that you are ill, you are spreading the viruses to other people, right? So it's not that we cannot fight it off, but in most cases, like, we don't do it fast enough, right? Which is what the vaccines are for. The vaccines train your body to do it. So when you get infected, you can make the antibody right away. Right? There is like very little wait time. We'll talk more about vaccines later. Okay, type of cells involved. Okay, can you give me like 2 cells that are involved in innate immunity? Make me feel a little bit better, please.
Platelets and white blood cells.
I see. I heard white blood cells. Yeah. What did you say, Tahib?
We have natural killer cells.
Yeah, the natural killer cells. Yes. And Mohammed, do you have a more specific example of
Platelets.
Oh, well, platelets is to stop the bleeding, right? We are specifically looking for things that fight off infections. Right? So like macrophage, right? And neutrophils. So these are some examples of cells of the innate immunity. Do they only eat a specific pathogen? No, they eat anything that's not supposed to be there, right? That's why it's innate. For adaptive immunity, as you will see, there are B cells and T cells. In terms of innate immunity, none for innate. Okay. Every time you encounter the thing again, tomorrow, if you touch a dirty surface and you eat chips with your hands again, you're gonna have to fight them off all over again. Like like the innate immunity doesn't remember what it fought off previously. Okay, but for adaptive immunity, there is generally long-term. If you had chickenpox before, you're not gonna get it again, right? Because you remember fighting the chickenpox off, so you are able to not catch it again. Sometimes it comes back as shingles, but that's like a different story, right? Any questions about innate and adaptive immunity? All right. Let's just do one poll question and then we're gonna take a break. I have a really long note that I need to give you. So it's probably a good idea to, take a break first. There you go. Actually, you know what? We cannot do this yet. I have to give you the note first. So why don't we just go ahead and take the break now? 5 minutes, and then we'll resume at 9:40. All right. See you guys in a bit. Okay, it's break time. Okay. Okay, so let's take a note together. All right. And this is gonna be a very long and a big note. Okay. And it's about antibody production. So just to give you a heads up, I think you should do the thing sideways. I'm gonna start here and then I'm gonna, do another thing here and then, go towards the bottom right. Okay, so that's how the note is gonna be. All right. This is antibody production. So, let's say we are infected by these red bacteria. And again, I keep on saying bacteria, but it would apply to other pathogens as well, right? But it's generally easier for people to visualize bacteria. So we have a bunch of bacteria and unique color for this note. Otherwise, it will definitely not make sense. Okay, just like a few colors, doesn't have to be fancy. Okay, so red bacteria. So how are we gonna clear them out? Well, we're gonna have to have this guy, right? This is going to be a macrophage. What is the macrophage going to do? Macrophage is gonna do phagocytosis, right? So it's gonna basically eat the bacteria. Okay. Let's just say, if this was a, not something particularly vicious, okay, and the macrophage will be able to clear it up, and then that would be the end of the story. You're not gonna have to make the antibodies and whatnot, okay? However, if the innate immunity is not going to be able to get rid of this bacteria, like the fever is not doing anything, the inflammation is not enough, the complement is not enough, then we have to trigger the adaptive immunity, right? And so one of the not unique, but like one of the special abilities of macrophage is that after it eats up the bacteria, it can do something called antigen processing. Okay. And essentially what that does is the macrophage will break down the bacteria and then it will put bits and pieces of it on the cell surface. Okay, so these are bacterial antigens. Of course, I'm simplifying the process, but essentially that's what's happening. Antigen processing involves breaking down the pathogen and displaying its antigen on the cell surface. Okay, so that's called antigen processing. It eats the red bacteria, it breaks it down, it puts it on the membrane. Okay? It's like it wants to show all the cells, hey, look what I just ate. Okay, this is the enemy. That's what it looks like. Go find them. That's antigen processing. Okay. All right. I'm gonna leave that for now. We're gonna come back to the macrophage later. Over here, we have, I don't know if you know what I'm drawing. That is the bone marrow. Okay. And bone marrow is where you get new white cells, right? As well as like other blood cells, like red cells and platelets, but we're focusing on the white cells. So of the different white cells that it makes something called naive T cells. Okay. You can like the proper way is to have two dots on the eye. I think it's like, I don't know, French or something, but it's okay. You can just write NAIVE, but naive T cells. Okay. So these cells are like T cells that are not activated yet. Okay. These not been activated yet. Okay. Each naive T cell has a unique receptor created through gene shuffling, right? I told you about gene shuffling earlier. It's like having a deck of cards, and then you randomly choose 10 cards every time. Those 10 cards are going to be different, almost every time, right? You have many different combinations, is what I'm saying. Right? So let's draw some naive T cells. That's a naive T cell, that's a naive T cell. And this is where you need the colors. Okay. So this first one, it has like a blue receptor. Now, of course, the receptor that I'm talking about is actually proteins, and it's actually the shape that matters, but it's much harder to draw the shapes in the note. So I'm just going to use color to represent them. Okay. So through gene shuffling, the T cell might have a blue receptor. Okay. Another one might have an orange receptor. Oops, where did it go? Orange receptor. Another one might have a red receptor. and another one may have a green receptor. Okay, so these are receptor for recognizing things. Okay, so at any given time, you would have a pool of naive T cells in the body, and they would have a different receptor for each of the T cells. Okay. I'm gonna pause there for a second, let you catch up, and please ask questions if there's anything I can help clarify. Okay. So back to our macrophage here. The macrophage is basically gonna go and talk to the naive T cell in a process called antigen presentation. That's what it's called, antigen presentation. Basically, the macrophage is gonna talk to the naive T cell. It's like, hey, I just ate this red bacteria. It's causing a lot of trouble in the body. Can you help fight it? Right? So it's gonna talk to the first naive T cell right here. Is this one gonna be able to help with the infection? What do you guys think?
No, it's not red.
Yeah, he's not red exactly. So like, oh, too bad, I can't help. Ask the next guy. All right, so then it's gonna ask the next one. All right, this is not gonna be able to help. Only the T cell with the correct receptor is going to be able to help to fight the infection, right? Now here is the thing, listen carefully. I said at any given time, you have a pool of naive T cells with different receptors, right? And these are generated randomly through gene shuffling. So it is entirely possible for you to not have the red one at the moment, just because like you're not like just by chance it's not there, right? So in that case, it will take a longer time for your body to make the antibody, right? You gotta wait for the red one to become available, right? But you're constantly creating new naive T cells, right? So the chances you will get there, but it's gonna take some time. Okay, so this one is gonna be the chosen one. Okay, the naive T cell with the correct receptor will be chosen, will be activated. It is the one. Okay, so antigen presentation involves selecting the naive T cell that has a matching receptor. So if the cell is able to do antigen presentation, then we call it an antigen presenting cell or an APC. Okay, so macrophage is an APC. Not all white cells can do this. Right? Neutrophils, even though they can do phagocytosis, they cannot do antigen presentation. Right? So it's not an APC in that case. So we only have one example, okay? The macrophage. Macrophage is an APC because it's able to present the pathogens. Sorry, the antigens, right? To the naive T cell. Any questions before I move on?
So all of those macrophage will present them, or only a few of them will present them?
Any macrophage that has eaten up a bacteria, they're gonna go present it. Right. So like, you know, you
Will it
I'm not
not be so many macrophage going to present because there are so many macrophage will be eating so many bacteria and
that's right, which is a good thing, right? Because you want to, you know, you want to select all these T cells that are able to help, right?
okay.
Yeah. So like, you know what? Maybe I'll not say this part. It's probably going to be confusing. Yeah, we'll just leave it at that. Okay.
After eating, they will they will go to bone marrow, and from there they will collect them.
Oh, not the bone marrow, but the T cells. Remember we talked about how the T cells go to the thymus, right?
Yeah.
Yeah. So that actually some of them happens there. Okay.
Huh.
All right. So the story is that we selected the naive T cell with the right receptor, right? So what's gonna happen next? What's gonna happen next is what we call clonal expansion. Okay, clonal expansion. Basically, the chosen one is going to do mitosis, and you're gonna have a bunch of them. Okay, you're gonna make many copies of the T cells. Okay, there we go. Which makes sense, right? Because it's like you can't just rely on one cell. You need like a whole army of them. We're not making the green ones. They're not gonna be able to help. We make the chosen one, the red ones. All right. So these cells they are going to differentiate. They are going to specialize into different cell types. Okay. So there are 4 things it can become. There are 4 things that they can become. The first one is called cytotoxic or killer T cells. Not to be confused with natural killer cells. Okay? They are both killers, okay? But this one is killer T cells. Okay? Or cytotoxic T cells. And so that's going to be like, you know, your T cells. And to help you remember it, okay, this one is going to be holding a sore, right? It's going to help kill things off. Right? So what does the killer T cell do? It will kill infected cells by poking holes in them. Okay. So poke at them, make them leak, make them die, kill the infected cells. Okay. So these naive T cells, right, some will become the cytotoxic T cells. Right? It's like different options, right? That they could become. If they don't want to become a killer T cell, they could become a memory T cell. Memory T cell. Okay. Memory T cell. Okay, this is like a thinking bubble. All right. So memory T cell will stay in the body. For a very long time. And be ready to kickstart immune response if there is a reinfection. Again, I love the chickenpox example because it's, it's the easiest one to visualize. If you had chickenpox before and you fought them off, you're gonna have a memory T cell somewhere in your body. If your kids have chickenpox and then the virus gets into your body, no problem. You're not even gonna have any symptoms because your memory cells will be like, oh, I remember that thing. We fought it off before. Let's just, get rid of it one more time. Okay. So by having these memory T cells in our body, we have long-lasting protection against things that we have previously fought off. Okay? So they could become killer T cells, they could become memory T cells. It's like you guys, after you graduate from the pre-health program, you can become practical nurses, you can become paramedics. There are different options. Here is a third option. Okay. They can become a suppressor T cell. Okay. When this is all over, when the infection is clear, you gotta tell these things to stop. Okay. That's what a suppressor T cell will do. All right. Once you clear the infection, once all the red bacteria are gone, you wanna stop the cells from dividing, right? You wanna stop the clonal expansion. Otherwise, you have uncontrolled cell division, which can lead to like blood cancer, right? So that's why we need the suppressor T cell to stop the immune response when infection is cleared. Okay. But don't worry, you already made the memory T cells. They will stay in the body. If we see this thing again in the future, your body knows how to deal with it. Okay. When is this not gonna end? Soon, okay, soon. There's one more option. One more option. Right? They could become a helper T cell. Helper T cell. Okay. Helper T cell. Actually, you know what? This one has a very big hand, like a helping hand. Okay, like I can help. I don't know. If you think of a better way to represent them, please let me know, okay? So what does the helper T cell do? Well, the helper T cell will activate naive B cell, which I will tell you what they are in a second. Okay? So all these cells, the killer T cell, memory T cell, suppressor T cell, helper T cell, these are called the effectors. Effectors of T cells. If the test asks you which of the following are effectors of T cells, it's these four. And then they are part of cell-mediated immunity. Okay, they are immunity that are carried out through cells mostly. Okay, so this here is it's differentiation, right? Okay. When a cell transform into one of these things, we call it differentiation. So there are 4 paths. So again, I want to take a small stop here, pause here, and just let you think about it. Anything you want to ask me at all?
What is cell-mediated immunity?
So like anything, all these activities that are carried out by the T cells, that's what we call cell-mediated immunity. It is immunity that is carried out by cells as opposed to carried out by antibodies. Okay, so cell-mediated immunity. All right. So let's go back to the top here. And we have this bone marrow thing, right? So other than naive T cell, we also have naive B cells that are being made. Okay, the B cells will stay in the bone marrow, but the T cells will go to the thymus, right? It doesn't really matter. Okay, so the naive B cells. Right? So again, you know the story now. We will have a bunch of naive B cells. Some will have the ability to make red antibodies. Some will have ability to make green antibodies, right? Some will have ability to make like purple ones or yellow ones. It doesn't even have to be the same as these ones, right? In fact, they probably likely not the same. Right? So each naive B cell has the ability to make a given, to make a certain antibody. And then it becomes like a familiar story now, I hope. The helper T cell, which is like this guy, right? It will help activate the naive B cell. So basically it will come over here and do another round of presentation. Okay, although that's not what it's called, okay, in this case, but it's same process. It's gonna basically, you know, we don't need any of these, they're not gonna be helpful, but we're gonna be selecting this guy. This is the winner. Right? That's the one that has the right antibody shape, and it's gonna be selected, and it's gonna undergo clonal expansion again. So you're gonna have a bunch of the B cells, and they will have the right antibody. Right? Almost there. And these guys can also differentiate. I don't know how much space you have, okay? But they can also differentiate. But they only have two choices. Choices are a little bit more limited here. Okay? They could become, okay, memory B cell. I'm not gonna write out the function. It's pretty much same as memory T cell. So you keep a copy of the T and you keep a copy of the B. Next time you get attacked by the same thing, you are so ready. Or it can become these cells that are called like plasma cell, which are like antibody factories. Okay, so they're gonna pump out a lot of antibodies. And more importantly, the ones that you need, right? So the plasma cell will mass produce antibodies. What will the antibodies do? Well, they can help neutralize toxin, neutralize virus, even though that's not what it's doing in this case. Form immune complex. Right. Actually, you know what? Let's put it there. Like clump bacteria together. Makes it easier to eat, right? To phagocytize. Also, it can help activate complement proteins. Okay, so hopefully all these things will be able to clear the infection, and the suppressor T cell will come along and say, "Ah, well done, good job. Infection's clear. Okay, stop dividing." And then we keep the memory B cell and T cell in the body, and it's done. So that is how antibodies are produced. Okay. So basically, to do a quick summary. Oh, sorry, not done yet. I forgot. Let's go back to here for a second. The plasma cell and the memory B cell. Okay, that's part of. These are the effectors of B cells. Naive B cell. Part of antibody mediated immunity. If you read this online, it's sometimes also called humoral immunity. Same thing. So summary. If you have the naive T cell, you are gonna need an APC, like a macrophage, to activate it. And then you will get, all the cells that we talk about. The cytotoxic T cell. You will get the memory T cell. You will get the suppressor T cell. And you will get the helper T cell. Just like the APC activates the naive T cell, the naive B cell will be activated by the helper T cell, which will become memory B cell and plasma cell. Okay. That's it. Any question?
Can you explain the plasma cell as a little bit again?
Yeah, the plasma cell is basically one of the things that the naive B cells can become. And the plasma cells are just antibody factories. They just keep on producing the antibodies that you need. In this case, the red antibody, right? So,
Thank you.
okay. hopefully that makes sense. Please reorganize the notes when you have time, and you know, write a nice copy and keep it next to you when you do the test. Okay, so back to this. You know what? Actually, I want to say one more thing here. So remember the RH sensitization thing that we talked about, right? It went through this exact same process, right? Except instead of having the bacteria here, you're gonna have the red cell with the RH on the surface. Okay, so like a macrophage will pick it up and then displace the RH on the surface, and so on and so forth. So it's gonna go through this whole process, and in the end, the person is gonna have anti-RH antibodies, right? They're gonna have memory T cells and B cells against RH in their bloodstream. That's why in that second pregnancy, right, the antibody is gonna be causing the problems. Okay, so that's that.
Professor, you have an application question that if the lady has already an RH and then second term is asking for the medicine, is it a
Yeah, let me get back to you on that one because I want to finish this thing first. But yeah, that's the application question in lecture 2, right?
yeah.
Yeah. All right. So, which of the following statements regarding antibodies is or are correct? Select all correct answers. Okay. That was a minute. Okay, let's see. We are looking for the correct statements. So antibodies are Y-shaped proteins. That's correct. Right, we've drawn it a couple of times already. You should remember it. B, antibodies produced against one pathogen will generally work against another pathogen. Not true. Will generally not work against another pathogen. The red antibodies you make are only good for the red bacteria. It's not going to work for like the orange virus, right? You need to fight orange virus. You're going to go through the whole process again and make, you know, orange antibodies. So generally, the antibodies you make for one thing is only good for that thing. But as you will see, there are sometimes exceptions. C, antibodies are produced by plasma cells. Yes, plasma cells are antibody factories. Antibodies are found in blood plasma. Yes, plasma is the liquid portion of the blood, right? That's where the proteins are going to be, including antibodies. Variations in antibodies arise from gene shuffling. Yes, that's how we can get many different combinations. All right, billions of different combinations. Okay. We did the antibody production note. I have another note for you. I'm sorry, there's a lot of notes and a lot of information. But before that, I want to do some questions just to see where you guys are at when it comes to vaccines. Okay, so people sometimes might have some misconception about vaccines, and I just want to see what people think. True or false? It is possible to get the flu from the flu shot. What do you think? Just choose. It's like there's no right or wrong. I mean, there is a right answer, but then you just choose what you think. It's what you believe in. Okay, and then I will tell you, explain to you what's going on. Can you get the flu from the flu shot? It's like same as asking, can you catch COVID from the COVID shot? Okay, I only get 8 responses. What about the rest of the class? What do you guys think? Okay, 4 more people. Are you guys there? All right, that's as much results as I'm gonna get. Okay, 75% of the class, oh no, so 67% of the class says that this is true. And 4 of you think it is not true. The truth, the correct answer is that is not true. Okay, you cannot catch the flu from the flu shot. You cannot catch any of the disease from any of the vaccines. If you're trying to get vaccinated against polio, you are not going to catch polio from the vaccines. Okay. I know it's hard to believe, hard to change people's opinion about this, but it's the truth. The flu virus is not even disease causing, right? Okay, in the vaccine, they have been weakened. Okay, it's called an attenuated version, so they do not cause disease. But some of you might wonder, how come I feel sick after I get the vaccine sometimes? Like if you get a vaccine, you might, feel the fever coming in, right? You might feel like you're under the weather for a few days. You have to understand when you inject the vaccine into the body, it is a foreign antigen. Okay, they take away the disease-causing part, but then nevertheless, you are still trying to introduce a foreign antigen, right? And so your innate immunity is responding, right? In the process, your body is trying to make the antibodies. And that's why you feel a little bit tired sometimes, and even, with the fever. That's the side effect of getting the vaccine, right? It's not the flu that's causing the problem. So in general, how does a vaccine work? Well, basically, let's say, you have the virus here. Well, let's go back to the other thing. You don't have to draw this out unless you want to. To keep it very simple, okay? Here is a virus, okay? And the virus has these proteins on it, like the blue proteins and then the yellow proteins, like that. Okay, let's say the blue one is the one that causes illness. It is possible to only harness the yellow parts and then introduce it into the body. So you will make yellow antibodies, but the yellow part is not the part that caused you to become sick. Okay, so that's why when you inject the vaccine, you're not actually going to catch the flu, right? So once you have these antibodies, the next time when you get infected by the real virus, you are ready because you have memory T cells and memory B cells for the yellow parts. So immediately those antibodies will neutralize the virus, and you will not get as sick. Okay, now that's how it works. You do not necessarily get full protection, right? it's important to understand the vaccine doesn't prevent you from catching the virus or catching the disease necessarily, but it causes your recovery time to shorten, and it minimizes your chance of becoming severely ill. Okay, you can still catch the flu, but you just won't be as sick. So so that's that. Okay. Any questions, comment at all?
Why the vaccines are age specific?
Why are the vaccines what? Sorry?
Age specific, like 6 months, 1 year, 3 years, like that.
Oh, that's just like the dose, right? Like, so like, because the kids are smaller, you don't give them like the full dose at once, right? Like it might overwhelm their immune system, right? Like they're not gonna, get hurt from it necessarily, but then like it might make them feel very tired, right? So like you want to, give smaller dose and spread it out further, right? That's essentially the main reason. Okay, let's try another one. Okay, I want to know what about this? This is one of those things that are out there. Okay, so like you might have heard that the COVID vaccines, some of them are mRNA vaccines. So people think mRNA COVID vaccines will alter your DNA permanently. What do you think about this one? True or false? What do you believe? Do you think this is true or false? Okay, anyone else before I close the poll? Again, this is anonymous. I don't know who chooses what. I am just curious what people think. Last call. Okay. Majority of you think it is false, and that's the truth. Okay, like mRNA vaccines will not alter your DNA. So basically, this is what happens. Okay, the RNA contains the instruction to make. Here, I'll do it on the other slide. Okay, so here is let's say this is a COVID virus, right? It's called the coronavirus because they have all these like spikes on them. Okay, they have all these spikes on them. So these are spike proteins. One of the first thing people did at the beginning of the pandemic was they sequenced the whole genome of the virus. So they know exactly what mRNA sequence is going to make those proteins. Okay. So what they did is they protect the mRNA in little oil droplets. Okay. This mRNA technology has been around for quite some time, even way before the pandemic. Okay, and then they will inject this into your body. Okay, they will inject it into your body. Once it's injected into your body, what happens is you will have an APC. Okay, it's not quite the macrophage in this case, but doesn't matter. It's the same principle. The APC. These mRNA will go into the APC, and then the APC will basically do translation. Remember last semester, transcription, translation? And it will put these spike proteins on the surface. Then the APC is going to do antigen presentation. Okay? And then it's going to identify the T cells and so on and so forth. And so you make a lot of antibodies in the end. Okay? The spike proteins don't give you COVID. It's just part of the structure of the virus, right? So when you do get the COVID virus, that's okay. You already have the spike, the antibodies to neutralize them. Okay. But what happens is the virus can mutate, so the spike protein can change subtly, so that previous vaccine no longer works. Right? So that's why they have to modify the vaccines, right? If there is like an Omicron variant coming out, they need to tweak the vaccines so that the shape of the spike proteins corresponds to the variant. And that's why you need to update the vaccines every now and then. And that's the same reason why you need to get the flu shot every year, because the virus mutates. Okay. All right. I hope this makes sense to you. But again, it's hard to change people's perception sometimes. Okay, but that's the truth. That's the scientific truth of what's really going on. Don't take my word for it. Right? Do your own research. Okay, how about the next one? True or false? Vaccines contain live viruses or bacteria. What do you think? Come on, let's pick up the pace a little bit, guys. What's your gut feeling about this? There's not much to think about, right? You either think it's true or not true. Okay, fantastic. I got my usual 13 response. Looks like you guys are half-half. It turns out it depends. Okay, it depends. Some vaccines use live viruses. Okay, like polio vaccines or even some flu shots, right? They have live virus, but it's important to remember they are attenuated. Attenuated. Okay, they has been weakened to a point where they do not cause disease. Okay, it's just like an empty shell, and sometimes it's just part of the virus, right? Okay, so like if it's part of the virus, obviously it's not alive, but sometimes they do use live virus and live bacteria, but again, they have been weakened to the point that they don't cause disease. Okay, using live vaccines are actually the most effective. They are the most immunogenic because it's the closest thing to the real thing, right? But it's not suitable for everybody. Right? People who are pregnant, for example, people who are immunocompromised, right? People who are undergoing cancer treatment, right? They are not suitable to get live vaccine. So the answer is true for some vaccines. Okay, depend what disease you're trying to immunize against. Okay, just a couple more and then we'll wrap it up. Some vaccines can increase the risk of autism in children. That's another common one. That's out there. You will hear people throw this around, if they are, anti-vaxxers, for example. But is it true? Okay, this is absolutely not true. Okay, there was one publication that showed that there was a link between vaccines and autism, but that study was actually fabricated. Okay, the results were made up, and it has since been taken off from the journal. And since then, there are so many other studies that try to show whether this is true or not, and all of them shows that there are no connection between vaccines and autism. But the damage is done. Okay, that false paper at the beginning is often used by anti-vaxxers as proof to support this untrue claim. All right, that is it for now. Now I do have one other note that I want to do with you before we go. Can you guys handle it without another break? Okay, hopefully. Okay, hopefully. So this one is called type of adaptive immunity. Oh, we lost someone. Type of adaptive immunity. So, new sheet, I'm gonna do a flow chart that looks like this, okay? If you're planning for space, that's what's gonna be happening. So let's start with the adaptive immunity. So we all know what that is now. This is specific. It's not right away, but you know, it's something that your body is able to develop, right, and offers you protection. So it turns out this can be divided into two categories: passive and active. Passive and active. Let's talk about what that means. In passive immunity, the host, like you are the host, okay? You are the person that's getting sick, right? The host receive antibody created by someone else. Doesn't have to be someone, like could be something else, okay? But the point is they receive it. Okay, so you don't have to make your own antibody. Someone's gonna give it to you. This is gonna give you immediate, but short-term protection. It's like cheating on a test. You will get the answer right away, but you know, you didn't go through the hard work of studying. You're probably not going to remember these things. So that's passive immunity, huh? Active immunity, the host makes the antibodies. That means you have to go through the whole T cell activation, B cell activation, clonal expansion, all the hard work. So that's why it is not immediate. But because you make the memory cells, it is going to be a long-term protection. Okay? That is passive and active. Okay. So for passive, we have two. Oh, sorry, I haven't really thought this out. But for passive, we have two types. Okay. We have natural or artificial. Sometimes artificial is called induced because you make it happen. Basically, if you are injecting something, then it's artificial. If it's just happening on its own, then it's natural. Okay. So natural passive. What's an example of that? Right. A mother can pass antibodies to her baby through the what? We learned about this before. Fill in the blanks for me. How does the antibody from the mother go to the baby?
Placenta?
Thank you, placenta. Right? But in this case, we're talking about like good protective antibodies, right? Not the anti-Rh stuff. Right? Like if the mother has anti-measles antibodies, that's going to go to the baby through the placenta. If she got the COVID shot, vaccines, the antibody is going to go to the baby. Okay, through the placenta. My sister was pregnant during the pandemic, like early on, maybe middle, and she was worried whether she should get the vaccine or not. So her doctor said, of course, get the vaccine. When you get the immunity, the baby will also get the immunity. Okay, but that's not long-term protection, right? The baby did not make the antibody itself. It received the antibody from the mother. So after the baby is born, those immunity will be there, but only at the beginning, right? The antibodies will break down in the baby, and eventually they will lose that protection. So what's one of the ways that the mother can help extend this protection? How can she continue to transfer the antibodies to the baby after the baby is born?
Is it breastfeeding?
Mother's milk.
Breastfeeding, that's right. Okay, so
Okay.
through the placenta or breastfeeding. There are antibodies in the breast milk. Okay. But like you cannot control what antibody to pass on, right? So sometimes like you might pass on certain ones and not the others. It's like, oh, it seems like a random process. Okay, so that's an example of natural passive. There is nothing unnatural about breastfeeding or, you know, transfer through placenta. And that's just what it is. Someone said, is insulin an example? No, not quite. Insulin is a hormone, right? So hormones don't really work that way. Not in the sense of immunity anyways. Everything okay about passive immunity? All right. Next one is artificial or induced. Okay, so this is an example of this would be a serum. Okay, serum. What is a serum for? A serum is basically antibodies extracted from immune individuals. Okay, so someone else have fought off the disease. We isolate the antibodies and then we mass produce that one. That's possible. Okay, like someone else fought off hepatitis. We get the antibody from them, we purify it, and we clone it billion times. Then you end up having a serum. Okay, a serum. So who's gonna need the serum? Okay, so this can be used for prevention of disease. Sorry, it's okay. E.g. Travel. Okay. I'm not gonna write the whole thing out. It's gonna like, you know, takes too much space. Okay, but listen, right? Let's say I have to go on to a business trip in 2 days. Okay, it's an urgent business trip. I gotta go in 2 days. The place I'm going has this disease. Okay, I have not been vaccinated for that disease. I don't want to catch it. Okay, it's too late to get vaccinated now, because even if I get vaccinated now, it's gonna take a week or so for the immunity to kick in, right? So we don't have time for vaccination. I'm not gonna order breast milk from Amazon or something, right? So what am I gonna do? Well, you can get a serum. Okay, so you can get antibodies from someone who has already fought off that disease previously. So I receive the serum, immediately I get the protection for that disease. I go on my travel business trip, I have the protection. and then I come back later, eventually I will lose that protection. It's good for like, you know, maybe a month or so, a month or 2 months. But once all the antibodies are gone, I don't have protection anymore. So next time, if I want to go again, I either get vaccinated early or I will need another serum. Do you understand? Okay.
Yeah.
It can also be used, for therapy. If someone working in healthcare accidentally poke themselves with a needle that they just pull out from another patient, okay, like let's say the person has hepatitis. After giving them a shot, you accidentally poke yourself, then it's a very scary situation, right? You're like, oh no, what if I caught the hepatitis? Okay, so you will receive a serum, okay, as both like a therapy and a prevention, because we don't know if you actually caught the disease, but if you did, then we would have the serum injected, right? Just in case you got it, inject the serum, and then it's gonna clear the virus before it causes problems, right? Okay. They have this for like HIV as well. If you think you got exposed, then there are shots and drugs that you can take to protect yourself in case you actually did it expose. Another example of this is rabies, right? Okay, what animals can spread rabies?
Raccoon.
Yeah, raccoons. Anything else?
Coyotes?
Okay, yeah. Anything that's more common, like, you know, that you can see people.
Dogs?
That's right, dogs, and bats, right? So, this was 2 years ago. 2 years ago, I was running as exercising, right? Like I run a couple of times a week. Okay, so I was running, and this person was walking two dogs. And you know, I was going one way, the dogs were coming the other way. They were on leash, okay. But I guess the road was reasonably wide, so I didn't go anywhere. I just, you know, run past the dogs. But the dog didn't like me, so it jumped at me and then it took a bite at my waist, okay. And so you know, I was shocked and I turned around and I said to the person, like, your dog just bit me. And then, you know, she didn't say anything. She just looked at me. So I guess when you run, like, your adrenaline is going, and then I guess the shock of getting bit, like, I got more adrenaline release or something. So I didn't really, I felt that it hurt, but then I didn't really pay much attention to it. Like, I was more annoying than like injured, right? So I was like, oh, whatever. Like, I said some not so nice things to the dog person, and then I kept running, right? And then when I got home, I wanted to take a shower, and then when I took off my clothes, I noticed I was bleeding, and I was like, oh crap, right? So the dog actually pierced the skin, and then I was actually bleeding. So I called my doctor, and I was like, you know, this is what happened. Should I come in for a rabies shot? Right? And then he's like, did you get the information from the dog owner? And I was like, nope. And he's like, well, in that case, you have to come in, right? Because supposedly if you guys ever get bitten by a dog, you know, try to get the information from the donor, in case, you know, if the dog shows any weird symptoms associated with rabies, ideally they will let you know, and that's how you know if you should be concerned, right? In Ontario, like, all the pets should be vaccinated, but you cannot be sure without the context. So the doctor is like, we don't know. We don't know if there's rabies or not. And once you start showing the symptoms, there is a 99% chance that you're gonna die from it. So better be safe than sorry, right? So that's what happened. I had to went in, and he actually had to order the vaccines from public health. Apparently, if you get bitten by a dog, they have to keep it in a record, right? So I received 4 shots on the first day. Right? Not only do I get the rabies serums, I also get the rabies vaccine, as well as the tetanus. Apparently, the dog saliva can carry tetanus bacteria, so I got that. And then I had to go back for a whole month, every week, to get additional shots of serum, right? As you can see, I'm well and alive. I did not get rabies, but that's what a serum is for. Okay. If you ever get into situations where you might be exposed to something, right? That's what a serum is for. It's not natural because you're injecting something, and you are not making the antibodies. Okay, someone else did already. So next time I get bitten by a dog again, I'm gonna have to go through the same thing. Which is why whenever I see dogs, like I'm a little bit scared of them. Like I you know, get out off the side road and run on the street. Next time I'm gonna get hit by a car instead. Okay, so that's passive. Now we're gonna talk about active. So active is also separated into two kinds, and I'm totally running out of space here. But the first kind is natural. Natural. An example of that is like my favorite example, right? Like chicken pox. Okay. If you caught it, right? If you got chicken pox, recovered, then you have protection. That's an example of natural active. I mean, it doesn't have to be chickenpox. It could be any other illnesses. All the diseases that you have overcame in your entire lifetime, now you have immunity for them. That's natural active. Okay? I don't know, like depending on your age, right? Like some for people who are older, like including myself, right? Like I got chickenpox the natural way. Okay, we didn't have a vaccine back then. But for my kids, they are vaccinated against chickenpox, right? It's actually it's like a routine vaccine in Canada. It's on the yellow card, right? But in the past, like according to my mom, she had this like chickenpox party apparently. Like she just like invite other people to come when I had the chickenpox. 'Cause her friends wanted their kid to also have the chickenpox, just to like get through with it, right? So that they don't have to deal with it in the future. And that's like
My mom
natural.
did the same thing too.
What's that?
My mom did the same thing too.
Yeah, yeah, the chickenpox
My
party.
uncle
Yeah,
had the chicken pox, and my mom made me play with my brother, my uncles. I got the chicken pox the same way.
right, exactly. Right. So it's a common thing to do. Okay, but for chickenpox it's okay because it's not a deadly disease, right? But for something like COVID, definitely don't have a COVID party. Okay, that's not a smart thing to do. Anyway, so that's natural. And then we have the active, right? The artificial rather. Artificial or induced. Active, right? So in this case, example would be vaccination. Right? Everything I told you about, right? Getting the flu shot, right? With a weakened form of the virus. And then when the real thing comes, you have the antibodies, you are protected. Okay? So there you have it. The 4 different kinds of adaptive immunity. So be able to identify examples. Like if I give you an example, you should be able to tell me whether it is artificial active or a natural active, artificial passive or natural passive. Okay. That is it for now. We still have like 20 slides to go or so, but that's okay. We'll do it next week. All right. And yes, I still remember about those application questions. We'll take it up closer to the testing. That's it for now. Thank you for coming and enjoy the rest of the week. Bye-bye.
Okay, so I have a question.
Yep.
Is the quiz not 27 marks?
Yeah. I lowered the average a little bit, right? The overall mark a little bit so that people get a little bit boost in their grades.
Oh, perfect. Thank you. I got like 25.25, so that's what I was thinking. Like, is it from 27?
Yeah, I made it out of 26 instead of 27, so everybody got a bit of a boost. Yeah.
Thank you. Thank
Yep.
you.
Okay, bye.